In this unit, we will be looking at the bones and the joints. The more obvious functions of the skeletal system include support, protection, and movement of the body. Other important functions include mineral homeostasis as a reservoir for calcium, red blood cell production in the red marrow, and triglyceride or fat storage in the yellow marrow. We can group bones based on the shape. The long bones are longer than wide and include the bones of the limbs, such as the humerus, radius, ulna, femur, tibia, and fibula. Short bones are more cube-shaped and found in the carpals of the wrist and tarsals of the feet. The flat bones have a relatively smooth and flat surface for muscle attachment, such as our cranial bones, sternum, and ribs. The irregular bones have more complex shapes and include bones such as the facial bones and vertebrae of the spine. Here we will look at the structure of a long bone. It is divided into three general regions. The diaphysis is the shaft of the bone, and the epiphysis is the end region of the bone, and the metaphysis is the region where the epiphysis joins the diaphysis. On the ends of the bones we have articular cartilage that you can see along here. This is made of hyaline cartilage to smooth the movement against another bone. The outside of the bone is covered with a tough membrane called the periosteum. In the center we have a hollow medullary cavity which contains the yellow fatty marrow, and inside is lined with a membrane called the endosteum. We can also see that there is blood vessels here called a nutrient artery that's going to bring a blood supply to the bone. Bone tissue is also called osseous tissue. The matrix is composed of 25% water, 25% collagen, and 50% mineral salts. The osteoblasts are the bone building cells that secrete the bony matrix around themselves. The blast ending is what indicates that it's a building cell. After they are enclosed in bone, they become osteocytes or mature bone cells. Cytes is the ending for cells. Here there is no cell division, and their purpose is to maintain the bone. The osteoclasts are cells to break down bone. They are located in the endosteum and contain lysosomal enzymes which break down the matrix. Our dense bone is referred to as compact bone. It is organized in repeating units called haversian systems or osteons. Down the center of these units we have a canal that runs longitudinally called the haversian canal or central canal. Volkmann's canals or transverse canals run horizontally and connect from the medullary cavity in the center to the haversian canals all the way out to the periosteum. These will contain arteries, veins, nerves, and lymph vessels to transport nutrients and waste throughout the bone. This top picture here illustrates osteons. You can see that there are multiple osteons next to each other. And down here you can also see you have an osteon here with the central canal running down the center with the blood vessels in it. And along here we can see we've got the transverse or Volkmann's canals that are going to run all the way from the medullary cavity out to the periosteum along the outside. This picture also shows the canaliculi, the lacunae, and the spaces the osteocytes will live in that we will talk about in just a minute. The bone is organized in concentric rings of hard calcified bone that get larger as you move out from the center, called concentric lamellae. The lacunae are the spaces between the lamellae that contain the osteocytes. In order to be able to pass nutrients to the cells that are not immediately next to the canals, we have canaliculi. The canaliculi are channels that radiate out from the lacunae and connect with each other. They contain extracellular fluid and processes or surfaces of the osteocytes, so nutrients can be passed out to the cells that are farther away. This slide shows an image of spongy and trabecular bone. It is arranged in a trabecular pattern with a lattice of thin columns of bone containing red marrow and osteocytes. This bone has the appearance of a sponge or Swiss cheese. Ossification is the process of forming bone. One way this is done is by intramembranous ossification. This process occurs in flat bones. Bone will form directly within the loose tissue between the membranes. The osteoblasts secrete matrix around themselves and become osteocytes in the trabeculae, and the vessels grow in the spaces in between. The other process is endochondral ossification. Here we begin with a cartilage model made of hyaline cartilage. This allows the models to grow more rapidly. As we have growth of the model, we have a pH change in the matrix that triggers a change in the solubility of the calcium salts, causing calcification or the deposit of the calcium salts into the tissue. This occurs in two different directions. In the metaphysis, we have a primary ossification center where bone begins to replace the cartilage of the bone, and this progresses inwards towards the center of the diaphysis or shaft. We also have a secondary ossification center next to the primary, which will proceed out from the center into the epiphysis. A nutrient artery feeds the primary ossification area. The secondary ossification area is fed by the epiphyseal artery. The epiphyseal plate is the space between the primary and secondary ossification centers that is the last place to be replaced by bone. This is easily seen in this fourth picture here and fifth picture. As a child, it is made of articular cartilage and becomes the epiphyseal plate when it's replaced by bone. After age 19 to 25, it is completely ossified and growth stops. So here it has gone from cartilage to being bone in this final picture. Bone is dynamic and changes with the stresses put on the bone. We have several hormones that are involved in this process of bone remodeling. PTH, or parathyroid hormone, operates in a negative feedback loop to maintain blood calcium levels. If the blood calcium is too low, PTH stimulates the osteoclast to break down bone and release calcium into the bloodstream. This also promotes the absorption of calcium in the gut and decreases the loss of calcium in the kidney to increase the levels in the blood. Calcitonin is produced by the thyroid and also is going to work on a negative feedback loop with blood calcium levels. When blood calcium gets too high, calcitonin inhibits the osteoclasts to stop bone breakdown. The rest of the hormones will promote normal bone growth. Thyroid hormone promotes normal bone growth as well as insulin from the pancreas. HGH, or human growth hormone from the pituitary, promotes growth of tissues including bone and stimulates the liver to produce the IGFs, or insulin-like growth factors. The IGFs also promote normal tissue growth and repair, which includes bone. Minerals are important in bone metabolism. Calcium and phosphorus are needed to form the bone salts deposited in the matrix, while magnesium is needed for support of the osteoblasts. Vitamin A is important for osteoblasts, and current recommendations are approximately 5,000 international units per day, since too much can slow bone growth and repair and may also cause birth defects. Vitamin C is needed for the collagen in the matrix. Vitamin D is needed to help with calcium absorption, but too much can cause bone loss and softening. Since bone responds to the stresses put on it, weight-bearing exercise stimulates bone growth. Most exercise has at least some weight-bearing component to it, except for swimming, water exercise, and bicycling. Strength training and weight training will give the greatest stresses to the bone, leading to bone growth.